Does anybody have any questions about these terms before we go on? Now, remember, I'm going to use these quite a bit throughout, not only in this chapter, but in subsequent chapters. So it really is important that you understand what they mean. So make yourself little flashcards or whatever you do to memorize stuff. I mean, obviously in school, it's not all just about memorizing, but there are some things, if you think about like your, I don't know what, your times tables, right? You just simply had to sit down and memorize those. And it's the same thing with this. You just have to become familiar with these terms. And the way to do that is to see them, to say them, to write them many, many times. That's where you get that familiarity. So make sure you understand what all of those means, but all of those mean. But before we go on, does anybody have any questions about any of those? Is there any confusions or things I need to clarify, as we start getting into the actual, what these terms are applied to? No, we're all good? All right. Okay, so remember what we were talking about the other day is the fact that Mendel, this Austrian monk, had collected tons and tons and tons of data where he had these really simple crosses between flowers. And what I mean by a simple cross is he was only looking at one trait. When we're only looking at one trait, when we're only looking at the flower color, you remember I said these flowers could differ in things like heights too, right? But if we're only looking at one trait, we're looking at what's called, I'm typing this in here, monohybrid. We're looking at what's called monohybrid inheritance. Mono means one, and the hybrid is the mixing together, obviously. So we're looking at how that happens. So later on today, we'll talk about something called dihybrid inheritance, where you're actually going to be looking at two traits simultaneously. So it's going to be a little more complex, but concept-wise, it isn't. So monohybrid trait. In this particular case, the monohybrid trait was the flower color. They were either going to be purple or they were going to be white, right? I'm just going to click the forward on my keyboard. And I just kind of went off for a second. I said it's always kind of interesting to find out the why behind the what. Why are there purple and why are there white flowers? Well, again, it has to do with that whole thing of enzymes, right? If you have that enzymes that helps you make that pigment, if you're a flower, then you're going to be purple. And all you need is one set of those enzymes from one parent. If either one of the parents lack that enzyme, then of course the flower is going to be white. So if you think about that's kind of the default color, right? All right, and as I said, Mendel did this so many times, it kept data in his little notebook over and over again. He began to notice a trend, and that really is the heart or the beauty of science, right? That's all the core of what's done in the scientific method, right? You make an observation, you make a hypothesis, you probably don't get it right the first time, so you go back and you make another observation, but what you're doing along the way is you're picking up on trends. You're saying, it's not working, but I do know that every time I do this happens, whether it's something you want to happen or not happen, right? You're starting to notice that happens. If you want to kind of think of it in a really simple term, we live with trends. In the summertime it's going to get warmer, so you start putting away your winter clothes, and in the wintertime the opposite happens. So trends are kind of like being able to, well, they're not kind of like, but they're being able to make predictions, which is what you're trying to do in science. Well, if I know that whenever I hit the wrong button on the garage door goes up or whatever, you're using the scientific method. So Mendel did that with his pea plants, and he noticed that when he did a particular cross, when he mixed particular types of parents together, that if he did this kind of cross the right way, what he would always get are these ratios that we talked about the other day, right? Make sure again you understand phenotype from genotype. Genotype you don't see, phenotype you see. So what he found out is he's always getting a 3-to-1 ratio. So 3-to-1. So if there were 100 flowers, then 75 of them would be purple and 25 would be white, right? It's not just 3-to-1, it's the ratio, 75% to 25%. Now, it doesn't always come out exactly that. I mean, you got to understand statistical noise or probability. I mean, the percentages might be, 74, 26, but it's going to bounce pretty close, and it's going to average around to that number of 3-to-1. And specifically, the 3 is going to be whatever trait you're looking at. In this case, it was flower color. The dominant trait, right? Remember, I always told you that in a genetic cross, there's going to be a dominant trait and a recessive trait. So the dominant trait, which is purple in this case, is going to show up 3/4 of the time, and the recessive trait is going to show up only a quarter. Same sort of idea with the genotype, same sort of idea with the ratio. A quarter of your offspring are going to be what we call true breeding, right? True breeding dominant. Remember, there can be two terms or two ways to use true breeding, because you can also call these guys true breeding. True breeding basically means that your phenotype matches your genotype. See what I mean? And that's not as significant with the purple one, but certainly with the white one, right? The only way you get a white one is if you're homozygous. So it's typically when we say something's true breeding, we're typically referring to this one. Because you don't really have to say true breeding for the white one. It's obvious because there's only one way to get a white one. So a quarter of your offspring are going to be true breeding, or there's the genotype terms, homozygous dominant. Half of them, a quarter and a quarter. Half of them are going to be heterozygous, and then just a quarter of them are going to be homozygous recessive, so the other true breeding. So that ratio, or those ratios, they're allowed them to make predictions. If I bred together, well, let's start easy. If I bred together 1,000 flowers, okay, so you guys do this for me. If you bred 1,000 flowers, or 1,000 flowers were born, how many of them, based on these ratios, how many of them are going to be purple out of 1,000? If you're pulling out your calculators for this, how many out of 1,000? How many purple flowers out of 1,000? Don't be shy, even if you've answered before. Ara, how about you? How many out of 1,000? How many are going to be purple? There you go. 750, right? I could give you any number. 1,000's a nice even number. What if I gave you a number that was a little less? Let's just pull one out of the air. Let's say 1,218 flowers. That's how many were born. How many of those do you think? How many would you predict are going to be purple if they were created in the way we've talked about Mendel doing? Now, if you don't know the number, you could also just type out the mathematical equation we'd use to figure that out. Is that right? Is that 75% of 1218? I think it is. It looks like it is. Is that what you, Marie, explain to, yeah, Marie, explain to the class what you did then. Type it out for me in the box there by what you mathematically did. Unless you did that in your head. Hold on a minute. You took 1218 divided by 4 times 3? No. Huh, it works. Holy cow. All right. That little math trick that I sometimes lose sight of, right? Here's how I would have done it. You got the right answer, so that's cool. I mean, I guess you're right. 128 divided by 4 times 3. Yeah, it's the same thing. But anyway, you figure out of 1218, you just multiply it by 75%, or remember, 75% is 0.75. I'll give you the same answer. You have to show me in class why that works. I'll have to think about that. That's neat. Anyway, use your method, obviously. But no matter what proportion I'm talking about, if I said, okay, how many heterozygotes are going to be born, then it's 2/4, you know, because the ratio adds up to 4. 1, 2, and 1. The other ratio, of course, adds up to 4. So whatever the numbers is going to be that many out of 4. So 2/4. That's why we say a half, obviously. Now, that's only going to happen. Remember, Mendel was able to watch trends, right? He noticed trends. Well, that's only going to happen if when he went out into the garden, the flowers that he picked to reproduce was a true breeding purple one, so one that had this genotype, and then a true breeding recessive, or I could just say a recessive, because again, that's redundant to have to say that it's recessive, or true breeding, obviously is. If anything else that he picked, he's not going to get that ratio that he got. So let's look at that a little more schematically so we can kind of use what we've learned, right? Here's the parental generation. As it says here, it's true breeding parents. Okay, so I'm going to fill in the genotypes. Remember, what we're looking at here is the phenotypes, what color the flowers are. All right, everybody agree? Those are the genotypes. And then out of the one that's purple, we have to figure out what type of gametes it can produce. Now, I don't mean what type of gametes in terms of male or female. What I mean is what kind of gametes in terms of the alleles they have. Now, one thing I do mean as it regards gametes is something we learned in meiosis, that gametes only have haploid, number of chromosomes. So instead of having 2 Ps for a particular trait, and this is something to pay attention to, this is something you can make a mistake when you're doing genetics problems. Just think about the biology. Those 2 Ps represent the chromosome information that's on the homologous and the or the paternal, and the one that's on its homologous pair, the maternal. So during meiosis, only one of those letters is going to end up in a gamete. Now, I always circle them when I'm doing genetics problems, so it helps remember what they are. Now, it just so happens this case that the parent producing the gametes, they only have one type of allele. So it's sort of a slam dunk thing. And then in the F1 generations, we do the same thing. Now, Mendel understood probability. And probability means that when you are trying to figure out the odds of something happening, okay, how many purple flowers are going to be born, you're really also at the same time figuring what are the odds that it's not going to happen. I mean, I could reverse that question. I could say, all right, with the previous slide, what are the odds that you're not going to get a purple flower? Well, that's 25%, right? Because that's how many white ones you're going to get. So in this particular case, if you're trying to figure out, all right, you got a gamete over here, and you got a gamete over there, and gametes get together by random. We talked about that in meiosis. But then again, this is kind of a simple one to figure out because there really is only one type of allele in each gamete. So all these purple flowers are going to be heterozygous, aren't they? They have to be. They get one allele, one letter, one allele from each parent. So that while that flower is the same phenotype as the parent above, right? Exactly the same, not a little lighter color purple, but exactly the same color purple, then there's no way to tell its real genotype. And we talked about that in class too. I don't know if that purple flower is homozygous or heterozygous. Okay, but as I said at the end of class, if Mendel had stopped there, we may never have really heard about him or talked about him, but he had the insight to go one step further. We've heard before that whole thing about it skipping a generation, right? It many times is something that appears to occur in twins. Like if your grandmother had twins, then there's a better chance. I don't know how much better chance, but that you'll have twins. All right. So he's looking at these two purple flowers here in the F1 generations, and he says, all right, I'm going to go ahead and cross those two together. This is Mendel's brilliance. Now it gets a little more complex, doesn't it? That's where you can use that thing I showed you on the board, the Punnett square. And the Punnett square is just an accounting. It's a way of making sure that you keep track of all the possibilities. That's what it is. It's a way to make, if you fill in the Punnett square, then you've accounted for all the possibilities. Then you can count them up. Because when you're counting up probabilities of something happening, if you miss something, then that obviously biases the numbers. So now these parents, they can make different kinds of gametes when it comes to the alleles, right? Because and probability says half of them will be a gamete with the purple allele, and half will be the gamete with the white allele. Now, of course, this is where the Punnett square becomes beneficial, because you've got to say, all right, for the F2s, if we want to figure out all the possible ways those gametes can come together, we make sure we don't miss one. So this gamete here, I mean, it could fertilize this one here or be fertilized, depending which one you're talking about. It could also fertilize that one, and then this other gamete could get into the mix as well, right? So we use that Punnett square, putting one row and one column for every type of gamete each parent has. I usually put the males as the column and the females as the row, simply because, I'm trying not to, I try not to make a mistake. I'm trying not to confuse myself, so I do it the same way every time. Each parent is heterozygous. And then simply, if you've done all that, and having graded, oh God, a few million genetics problems throughout my time here, I can tell you this is where students often make a mistake. Right there, figuring out what the gametes are supposed to be, figuring out what the row and column headings are. So that's the part you really got to work on when you're doing your genetics problems, because the rest is just a 3-year-old can do. You just copy. This box right here represents one possibility for an offspring, a homozygous dominant purple one. These two represent purple flowers, but they're heterozygotes, and the only white one, the one that, you know, skipped a generation, is going to be down here. So now where do we get that 3 to 1? Well, out of 4 possibilities there, 4 because I just wrote them in there, 3 of them are going to be purple, or 3/4, and 1/4 is going to be white. And the same thing with the genotype, right? So 3 to 1 and 1 2 to 1 are 2 very important ratios that you need for making these kinds of predictions and understanding the way it works, right? Your phenotypic and your genotypic ratios. Phenotypic on top, genotypic is the 1 2 to 1. So make sure those are very, very, very firmly ingrained in your mind. Now, these ratios, these 3 to 1 ratios, these 1 2 to 1 ratios, they would really only be significant to purple or pea plants if they only happened in pea plants, but see, they don't. These ratios even happen in human beings, given that the trait that's being inherited is like this one. It's a monohybrid trait. Human genetic traits, well, in general, organismal genetic traits are usually a little more complex than that. Mendel was astute enough to have figured a trait, or to have used a trait or traits that were relatively simple in these organisms. Like, for example, if he looked at seed color or he looked at seed shape, first of all, he picked things that were very different. You know, it wasn't like yellow and greenish yellow. You're like, I don't know, does it look green? Does it look yellow? It wasn't like it was round or kind of wrinkled or not kind of round. These things were very distinctively different, yellow and green, round and wrinkled. And look at the ratios he got for those, right? When he crossed those together, those were the numbers he got, the 6,000 to the 2,000, and then the 5,400 to the 1,800. But look at the ratio. So even though he's moved on to other traits, right, and there are even other traits that he looked at in this particular species, like he looked at the shape of the pod, which he called either inflated or constricted. He looked at the color of the pod. He looked at where the flowers are. Axial flowers, not that this is a botany class, but leaves, stem, and leaf. Axial flowers come out of here. Terminal flowers grow at the top. 3 to 1, 3 to 1, 3 to 1. Stem length 3 to 1. It was all 3 to 1, right? Over and over and over and over again. So Mendel probably made a lot of money off of his beer-drinking monk friends, telling them, yes, you cry. Now remember, the stipulation that Mendel had to make if he was going to make a bet like that in this monohybrid, if he wanted to make sure he's going to get something pretty close to 3 to 1, he had to make sure whatever the parents that were chosen to him to prove this bet, both of those parents were heterozygous. Okay, so like, for example, pod shape. The inflated is the dominant. Remember I said we usually symbolize the dominant allele or the dominant trait by using its first letter and capitalizing it, and then the recessive trait is the same letter, less confusion that way. Now this isn't a hard and fast rule, so if you see alleles referred to in other ways, don't get confused, but typically this is what it is, and constricted would be a little lie. All right, so he had to make sure that in the bet, if he was going to use the pod shape, that the parents that were crossing were both heterozygous. So if we kind of use just X as a general thing, you're only going to get the 3-to-1 if you're crossing these kinds of parents together. They both have to be heterozygous. If you have a homozygous and a heterozygous, and you get a different ratio. You get a completely different ratio. All right, now this last slide kind of puts together just with some numbers for you. It's the same idea again. The reason why you have to put this Punnett square together is because sperm and egg, or the gametes getting together, is random. It's like that dude over there on the right flipping the coin. If you flip a penny, a fair coin, they say, half the time it'll come up heads and half the times it should come up tails. Now, it may be 51-49. If you did it once, obviously you're not going to get that. But if you did, the more times you did it, the closer it's going to be to. That's why Mendel did it, 6,000 times for some of those organisms that we were looking at in the other two slides. This became known as Mendel's law of segregation. And Mendel's law of segregation, laws in science are more like gravity's a law, right? It's like maybe I guess a theory would be a good way of saying it. The law of segregation, what that basically says in Mendel's genetics work, is that the gametes segregate from one another. In other words, basically he's saying what happens during meiosis, right? The chromosomes split apart. No matter what allele you're talking about, in this case it's the flowers, but the big P and the little p up there at the top, or I'm sorry, the purple flower, the big P, those are on different chromosomes. That's why there's two of them. He had two parents, and meiosis makes gametes with half. And so Mendel says because that happens, because they segregate, they separate apart from one another. They don't stay together unless rare instances where that occurs. You have to use that Punnett square to figure out all the possible ways they can come together. All right, and once again, in red, I put the ratios to remember, or to remind you how important those are. Now, one of the things I want to talk about before we leave monohybrid, first of all, does anybody have any questions on that before we go on? So as you're working genetics problems, keep that in mind that you're looking to get those ratios. Did I post those genetics problems online for you guys? Let me see. I have a set of genetics problems. They're not graded. They're the problems as well as the answers. Trying to see if I put those on the chapter. Let me see. Chapter 14, they would be in Lectures and Resources. Yeah. If you look in chapter 14, Lectures and Resources, there's a PDF at the bottom called Practice Genetics Problems. Do those. The answers are there as well. But do those, and if you feel comfortable with those, then you're ready to do these genetics problems on the exam and other places, okay? Now, some of the stuff that's on there, if you download them today, we haven't talked about yet, so don't worry. But we have talked about monohybrid crosses, so you can do those. That's the first set. Oh yeah, I wanted to talk to you guys about this whole idea of true breeding. Now, true breeding, if you're white, is easy enough because the flowers have to be homozygous. Recessive in order to be. Remember what I said true breeding means? Your genotype matches your phenotype. You see, the heterozygous one, the purple flower, that's not true breeding. That flower has a white allele. It doesn't have any white color, but it has the white allele. But the purple flower up there, the homozygous dominant one, that's the one that's true breeding. Now, in genetics, that's the unknown, you see. Even when you take my Bio 2 class, and we talk about this thing called the Hardy-Weinberg, it's a way that you can computationally figure out if species are evolving. But the question in the Hardy-Weinberg is the same question that you see here. If I go out into the garden, or maybe Mendel had, and I pick a purple flower, it could be either one, couldn't it? So I got to be real careful if I'm picking those two purple flowers to win my bet with those monk friends of mine, aren't I? Because I could go pick two. Now, if I picked a purple flower that wasn't a heterozygote, made a mistake, and that first flower was not a heterozygote. The second one was, but they were both purple. Am I going to get any white flowers? Am I going to get a 3-to-1 ratio, or what kind of a ratio am I going to get from that? Am I going to win my bet? Do a Punnett square if you need to. You can draw one out real quick. The question is, are you going to get any white flowers in the F1s? Any white flowers? Any. 1, 1,000. Get it again. Take a look at it again. Remember what happens. Those are diploid up above. They have to become haploid. You see any white flowers in there? See any white flowers? White flowers? No, none. None. You're not going to get any white flowers out of that. That's right. See how quickly I did that Punnett square? Boom, 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 right? There's your answer. So if you're sitting there going, wow, do a Punnett square. Take those letters, split them apart, make one a column heading, make one set the row headings, fill in the blanks, and count them up. You can quite quickly see in the 2 seconds it took for me to draw that, there's no white flowers down here at all. They're all purple, so I'd lose my bet, wouldn't I? All right, so back to this idea. How do you know that it's this one or this one? Well, there's a way to do that. It's called a monohybrid, aptly enough, a monohybrid test cross. So we're testing. We're doing a cross. When you're breeding organisms, that's a cross. And we're testing. We're testing this question right there. Okay? We're trying to figure out if this organism, going back to that true breeding word again, if this organism, this purple organism, is true breeding or not. And if you take a look down below, it's got basically a what-if kind of scenario. If you follow on the left-hand side and you say, okay, it was true breeding, then you can see the little Punnett square they put together. Okay? So you can see that now what they've done, don't get too confused about what they've done with the Punnett square. In this particular case, they put the white flowers, the column headings, and they put the purple flowers, the row headings, the one on the left. I'm looking at this one right here. Whoops, can't see my pen. Looking at that one right there. Regardless of the way you put them, it is a square, of course. Regardless of the way you put them, inside you're going to get all purple flowers. So Mendel would have done that. He would have picked a purple flower, and he would have bred it with a white flower over and over and over again. Now, just for a second before I go on, why did he pick a white flower? It was very specific. Whenever he did a test cross, no matter what trait he was talking about, the flower he crossed the unknown to was a homozygous recessive or recessive. Why was that so important? Heck, that's worth a point on the next exam to the first person who can answer that for me. So maybe that's what you guys were waiting for early when you weren't answering. It's like my dog, like Cooper when he comes in from the house. He won't. Carry on. Go on for the point. You're on the right track. Go on, though. It was a control. How was it a control? Someone give her a hand. How was it a control? Okay, you guys are being vague. Tell me very specifically how the white flower acted as a control. And if this is really just hitting a wall with you, think of the alternative. Think about whether if that had been a purple flower. Would he been able to answer his question about the flower on the left? You have the right idea that it is a control. And remember, the definition of a control is you keep only one variable. You're only testing one variable in a control, right? What's the variable here that's being tested? What's the question that's being asked? There's a question mark on the page. What's the question that's being asked? Come on, guys. in a monohybrid test cross, we're testing for something. No, the question is we're trying to determine something, right? We're trying to determine this right here. That's the question. That's the variable. That's the thing that we're trying to figure out. Is it one or the other? Is it variable, right? That's the thing that varies. So we're not going to add another variable to cross it to. In other words, we're not going to cross a purple flower to another purple flower, because guess what? That could be this, or there's the variability part. It could be this. So you're going to pick a flower, you're going to pick a control that is not variable. The white flower, you know what its alleles are. Does it make sense? So if we go back to that general way of referring to this, if you're trying to figure out this organism here is true breeding or not, and I leave the blank because that's the question, right? Is it a dominant or a recessive allele? You're going to cross it to something else that has alleles you already know. Now, the nice thing about a monohybrid test cross is Mendel, as I said, if it was true breeding, you got all purple flowers. Now, he didn't just do it 4 times, because someone could say, well, you can flip a coin 20 times and get 20 heads, and that'd still be a good coin. Maybe just by luck you got 20 heads, and on the 21st time you would get a tail. So in statistics, there is always that argument. But if you made thousands of flowers and they always came out purple, you know, the odds of the 1,000th flower coming out white are pretty minimal. But more importantly, well, as importantly, when it wasn't true breeding, when it was a heterozygote, he could predict the outcome. Half are going to be purple, half are going to be white. He got a 1-to-1 phenotypic ratio. So he could tell you, not only if that organism is true breeding, I can tell you you'll never get a white one. But if it isn't true breeding, I can tell you out of 1,000, 500 will be purple and 500 will be white. Boom, he breeds them together. He gets his ratio one way or the other. There are either no white ones or half. And he's a happy monk. Right? See how the beauty of numbers, right? You can predict things with that. You can establish things that are going on biologically. This just substantiates his idea of the law of segregation too. Those alleles are separating. And you can show how they separate, and you can prove that that's how they separate, because the results you get are either all purple flowers or half of them are purple and half are white. Remember, the idea behind science is to collect enough hypotheses, test enough hypotheses, develop a theory, and then take that theory and use it to make a prediction. That's really what helps support your theory. Okay, now we're going to go on to something called dihybrid inheritance. And in dihybrid inheritance, what we're doing here is looking at, this is still pea plants again, we're just looking at their seeds now. But it doesn't matter what traits we're looking at of those 2 slides that we went through. What was there? 7 traits, pod, shape, flower position, all that sort of stuff. No matter which one we looked at, what we're doing now is we're saying, how is it inherited when we look at 2 traits simultaneously? So now it's important that that seed up at the top is yellow and round, or the other one is green and wrinkled. In other words, it's important what color it is, and not or, and what shape it is. Okay, so it's important what color and what shape. So that, of course, complexes the things up a little bit. You notice, for example, the gametes down below for the pea generation. For one thing, if you look in there, now we got 2 letters. You think, well, wait a minute, I thought we're only supposed to have one letter per trait. This is 2 traits. Color is a trait, and shape is a trait. So you're going to have a Y, that's the allele for the color, and the R is the allele for the trait. By the way, in this one more time, the capital Y means that yellow is dominant and round is dominant, and then the opposite for the other 2, right? Now, in the F1 generations, you cross those together, and guess what? You don't get any green ones at all. And you think, okay, well, the green disappeared, right? Mendel goes off about his monkey, never does anything more beyond that, and we may never talk about him. But he didn't. He did this, just like he did in the monohybrid. He took 2 heterozygotes and crossed them together. And he ended up getting the ratios down below. Now, he had 2 hypotheses. He said there was one hypothesis based on dependent assortment, and one was based on independent. Now, this is the one that was substantiated. It ended up establishing his law of independent assortment. Now, here's what that means, independent assortment. Independent assortment says that these alleles, the Y's, the big and the Y, and the R's, the big R and the little r, each of those has an equal probability of being in a gamete with the other one. So the big Y has just as much probability of being a gamete as and the big R as the big Y and the little r, which has just as much probability, oh, there's a letter missing there, as the little y, it's just down below, and the big R. And those all have the same probability of having the little y and the little r. Now here's where the difficulty and the confusion and where the mistakes are made in dihybrid crosses when students are doing this. So let me remind you of something that you learned back years ago in math when you were learning how to do this. Oh, let me get my pencil. There we go. What's that called when you expand that out? You're expanding a what? I know you guys have had math. A plus b squared. Well, what's a plus b squared equal? If I wanted you to do that, what's that equal? Gosh, that's at least a squared plus b squared. You got part of it. Part of it. If you have a pen, you can draw on the screen, since I know it's a little hard to make superscripts in the chat box. Does this help? Does that help? What does foiling stand for, everybody? What's FOIL? What's it stand for? It's an acronym, right? What do the letters stand for? I know you guys must remember this because I know you've taken math. We're not going to have much time at the end to review if we're having to go through this. So there you go. Thank you. Yes, first, outer, inner, last. Marie, that's exactly right. That's how you do that. A squared plus b, 2ab plus b squared. Now, you're thinking, what in the heck does this have to do with what he's trying to teach me? Well, I'm trying to show you that Mendel's law of independent assortment says that you can use the foiling technique to make sure that you obey his law. In other words, you can use the foiling technique to make sure that you've arranged it up or you've accounted for all the ways those gametes can get together. Let me show you what I mean. First, right? One gamete can contain the first letter from each allele. Outer. Now, this time you're not going to add them, okay? But you're just going to use the words outer. The outer gametes can form an allele. Oh, I'm sorry, the outer alleles can form a gamete. Said that backwards. The inner alleles can form a gamete. And the last can form a gamete. So when you're doing dihybrid genetics problems, if you need to figure out all the different ways that gametes can be formed, and the organisms that you're talking about are what we call, here's a cool word, doubly heterozygous. Notice, doubly heterozygous because of the fact that they're heterozygous for both traits. So now we mix up the terms a little bit. Organisms could be doubly heterozygous. They could be homozygous and heterozygous, right? They could be homozygous for the first and heterozygous for the second. They could be doubly recessive. Okay, so that's where they got these row and column headings that you see there. That's how they figured it out. They foiled it out that way, and that accounts for all the possibilities. And then what do you do once you get to that point? You fill it in on the table. So sometimes what we do in a hypothesis and testing in science is we come up with what we think is the right answer, and then we also say this is what will happen if I'm wrong. Or sometimes we don't know which one. We come up with two hypotheses. His hypothesis of independent assortment was mathematically substantiated. He said that if I do it this way, that in the offspring, the F2 offsprings, now we don't worry about genotype ratios here. There is a genotype ratio in dihybrid crosses, but we're not worried to count them up because we're really, in this point, only going to focus on phenotype. And sure enough, he says, if I cross those together, that's the phenotypic ratio I should get. Now, you're thinking if it's 9 over what, add them all up, right? It's 16. So what he's saying is 9/16 of them, which is about 315, if you look down below, will be yellow and round. 3/16 of them will be dominant for one trait and recessive for the other. So it'll be green and round or yellow and wrinkled. And then just 1/16 of them, the one down here on the lower right, that's the one that's going to be both recessive traits, doubly recessive. Now, what his law of independent assortment, the way it was substantiated here is he said you have to basically FOIL out all the gametes because all of those are possibilities. Those gametes, those Y's and R's, can go off and form gametes in all those possible ways. Remember what I said about statistics and probability? It's about accounting not for just the way things happen, but the way things don't happen as well. So you got to keep that in mind, all right? So now you can work those dihybrid problems on the sheet as well too. So okay, 10:15. Yeah, we got. I tell you, I'll stop lecturing 10:30. 10:30. Okay, and then we can do a review if you need to do that. All right, down below where I said linked genes, certain genes are linked, like blonde hair and blue eyes. Certain characteristics we see in organisms are very often together. Folks with blonde hair often have blue eyes and vice versa. In fruit flies, it has to do with their eye color and their wing length. And the reason why that happens is those are kinds of, those are genes that are very closely, they're very close to one another on the DNA. So what happens is when you chiasma, when they cross over, those things are very often inherited together. So when we're looking at dihybrid inheritance, if we were looking at blonde hair and blue eyes, the two traits, we'd get some funky answers. We would get answers more like this. Because when you inherit blonde hair, you usually inherit blue eyes too. But those, of course, are the exceptions, okay? And everything has exceptions. We know why, though. It's not exceptions like we can't explain it. We know why, simply because they're close to each other on the chromosome. So when the homologous chromosomes cross over, these things often come over as a chunk. Okay, any questions on that? All right. Take a look at this. Now, you think you got everything figured out? Here's where Mother Nature throws us a curveball. There's something called incomplete dominance. And what incomplete dominance says is that the intermediate phenotypes, this is the intermediate phenotype. Between, in this case, I don't know what flowers they are, snapdragons or 4 o'clocks or whatever the book says they are, what kind of flowers. But there's red and there's white. Now, red is dominant. Okay, now, again, don't get lost in the symbology. As long as you know what the symbology means, in this particular case, the scientists have decided to call the alleles capital C with a superscript capital R for red and W for white. So they're going against that, that uppercase and lowercase lettering thing. It's no big deal as long as they tell you what they mean, right? So the red one on the left is true breeding, right? It's homozygous dominant, but incompletely dominant. And the one on the right is homozygous recessive. Now, here's, for every rule there are an exception. In this case, if you look at Mendelian-wise, if red is just completely dominant, and not incompletely dominant, but dominant in the traditional sense, what color flowers should we get down here in the F1? What color flowers should we get in the F1s if red is typical dominance, not incomplete dominance? Yeah, we ought to get red ones, right? But we don't. We get pink ones. And that's even odder because we're thinking, wait a minute, years ago they used to think that inheritance, they called it the blending theory of inheritance, which was, it was basically a tall parent and a short parent have a medium-sized child. And while that plays out very often that way, that isn't typically the way it works. Now, a lot of the early research that was done in biology, like Mendel's work and genetics, was done with plants. So So there were some geneticists out there that were kind of stumbling upon that some plants, a few plants, kind of actually do the blending thing, right? Red and white is pink. But this does not go against what Mendel said, and let me show you why, because there's a numbers thing again. If you breed the two pink ones together, you can make a prediction on the ratios. And specifically, the ratio that's really important is the genotype ratio. Look down below. How many red ones are there? And then there's two pink, right? And then there's a white one. There's that ratio, right? That 1:2 to 1. Now, this time we're talking about phenotypes. There's one red one, half of them are pink, and one's white. But at least when this was done, it showed that, oh, Mendel's right. It's just that these organisms are different. When they are the intermediate phenotype, or when they're heterozygous, like the red and the pink and the white, they're going to be the intermediate phenotype. Again, that happens in some organisms and some genetic traits. It's typically the other way. Mother Nature just likes to keep things interesting for us. All right, now, so far what we've been talking about was two alleles, right? The trait is flower color, it's purple and white, or it's constricted or inflated. Earlobes are either attached or they're not. Remember I said we made it easy there at that time. Most traits out there are controlled by multiple alleles. So there may be a red and a white and a purple and a green and, you know, whatever the trait you're talking about. There's many different choices. Not only two, there could be 3, 4, there could be dozens of them. An example of multiple alleles, one that's simple enough that we can explain without just going crazy needing a computer to do it, is the alleles for blood typing in human beings. In human beings, blood typing is called ABO. Now, that's not for the phenotype. That's not for the blood type. Because you remember blood types, there's A, B, O, and AB, isn't there? It's a rare one, but, okay. So the alleles we have, there's not 2 alleles, there's 3 alleles. And not only to complicate it that way, 2 of the alleles are dominant, all right? So in this case, we have something that's called codominance. Not incomplete dominance like in the flower, but codominance. See, again, they're just using an I here. The A allele is dominant, the B allele is dominant, the O allele is recessive. Now, the basis behind blood and why it's so important to type blood and why it's so important not to mix blood together has to do with these markers that are on the outside of the blood, these carbohydrates. And if you're an A allele organism, you make carbohydrates, they don't look like a triangle, but you follow the idea. You make one type. And if you're B, you make carbohydrates that are of a different shape, and if you're O, you make none. Now, these carbohydrates are identity markers. They're on the outside of the red blood cells. So if you're A, then you have the triangular ones, and if you're B, then the circular ones. I'll talk about the AB in just a second, though. But go back to the A. Notice you can be A 2 ways. Right? Homozygous and heterozygous. You can be B 2 ways. But now to the codominance. If you have both dominant alleles, you're not going to be in between this time, right? You're going to share it. So you're going to be codominant, and that's where AB comes from. Now, the O person there, they have no carbohydrates. The basis of not messing up or blood typing is that if you're A, you have if you're an A type, if your blood type is A, your body has antibodies to B. So if you put B blood into an A person, the antibodies attack it and they cause it to clump. It's called agglutination. They clump, and your blood clumping is not good. If you're AB, in the case of AB, you have no antibodies because you're not going to attack your own red blood cells. So AB can basically be a donator to everybody. And in the case of O is going to be the, O is the universal donor. Right? O has no markings on the outside, so no matter what antibodies you get, it's not going to attack it. AB is the universal acceptor. You just can't donate to anybody because if you have A or B antibodies, it'll attack it. And we could use this very simply for blood typing. Now, the thing about blood typing, like in the case of maternity suits, is blood typing is good at eliminating who the father is, but not so good at telling who is the father. And let me show you what I mean by this. Let's take a woman who has type, let's say she has type O blood. She's homozygous recessive. And by the way, just because you have just because you're the recessive phenotype doesn't mean you're in the smallest proportion. Many times, in the case of, like, for example, blood, a large proportion of the population is the type O blood. And let's say that they have a child who's type B blood. And for whatever reasons, we don't know who the father is, we leave the blank. Now, she claims that this guy she met, or this fellow she knows, or whomever, right? She claims that he is the father. And when they take him to the doctor and they type his blood, he's got type O blood. Now, you be the doctor. Answer the question. Is he the daddy? Is he the father of that child? The mother's on the left. This is the suspected father on the right. He's also type O. Is he the father? Anybody? Is he the father? There's no way, right? Okay, now, one thing we do know about the father, though, right? Whoever the father might be. What's the one thing we know about the father? I mean, we know it's not this one, but like I said, so we exclude this guy. But what do we know about the father? Whomever he may be. Sadie? Marie, Kelsey. What do we know about the father of this child? He's got to have, well, I don't know about recessive, but he's got to have a B allele. He needs to have a B allele. Whatever, he could be homozygous dominant, he could be this one, or he could be this one. He could even be one of these guys. He's got to have a B allele somewhere. So you see what I mean by a blood typing will exclude? They excluded the type O guy. He can't be it. But now who's the father? Any male out there with B or AB blood. That doesn't exactly narrow it down too far, does it, right? All right. Some questions on that. Let's see how many more slides. We can do one more at least. All right, pleiotropy. Pleiotropy is defined when one genetic trait influences many phenotypic traits. All right, so having one genetic trait influences the organism's body in many different ways. For example, in sickle cell anemia, which we talked about, having abnormal hemoglobin. Now, that is a single genetic trait. Either have it or you don't. So if you have the sickle cell anemia genetic trait, you exhibit pleiotropy. And the reason why you exhibit pleiotropy is this is all the phenotypic results of having that genetic trait. Phenotypic results, phenotype doesn't have to be good. Phenotype doesn't have to be benign, like you're purple or white or green and red. Phenotype could be that you have these traits of sickle cell anemia. So phenotype doesn't have to be a good thing or a benign thing. It can be a bad thing. So when we have one genetic trait that controls many different things, we call that a phenotype. All right, we call that a phenotype, and one gene controlling many phenotypes is a pleiotropy. All right, now, the opposite of that. The opposite of that. So now, in this particular case, what I'm going to be talking about, well, now let me show you one more example of pleiotropy. I just forgot to put this slide in. This is a new slide. This is kind of neat. Let me show you an example of pleiotropy. Pleiotropic genes can be really powerful. This will be my last slide. In the phenomenon biology known as epistasis. Epi is beyond, above, and stasis is like homeostasis, the way things are supposed to be. In epistasis, what we do is we have a gene that masks. It masks, M-A-S-K-S. It masks the expression of another gene or genes. I like to think about a gene, an epistatic gene, is one like when you take a big math problem, you know, all fancy stuff, and you multiply it by 0, it's always 0. That's kind of what an epistatic gene does. So look at these Labrador retrievers. We're going to talk about how we get different colors of Labrador retrievers. So up above, what you see is the Labrador retrievers that are doubly heterozygous mate, and so we got the square down below. Let's take a look at how this all works. So I'm going to show you the epistatic part. In this particular case, the gene is the epistatic gene is E. B is the color gene. All right. So if you're homozygous, you're going to be a black Labrador retrievus. If you're heterozygous, if you have one big B allele, then you're going to be black. If you're homozygous recessive, you're going to be brown. Simple enough, right? Big B black, little b brown. Now, here's the thing with E. If you're homozygous dominant for E or heterozygous for E, E being the epistatic gene, the masking gene, you're fine. No big deal. It's like nothing happens. If you're homozygous recessive, and a lot of times genetic disorders are epistatic, bad things, and oftentimes Mother Nature, they're set up as being recessive disorders, right? The traits you wouldn't want to be passed on. But look at this particular case, and this isn't a bad disorder for labs, but look right here. When you're homozygous recessive, that E gene, since you're homozygous recessive, totally ignores what's going on in the B gene. You see the one dog there that's golden? It's big B, big B. It ought to be black. And the other two ought to be black, and the one on the lower right ought to be brown. But because there's this epistatic gene, then it masks the expression of all the other genes. Albinism in human beings is an example of an epistatic gene. If you're an albino, you inherit the albinistic gene, the one gene that controls many traits: white skin, bleached out hair, the iris of the eyes, that sort of stuff. Okay? All right, let me see where I am in my progression here. Yeah, we can stop there. That's fine. Let me, no, you know what? I'll leave the recording on. All right, who's got questions for me on the exam that we're going to have on Monday? Same sort of format, same idea, upstairs, on the computer. Nothing's different. I don't know exactly how many questions about. Yeah, there is one. It's a compare and contrast one. I'm not going to tell you the topic. I think it's probably pretty obvious. I have more than two topics I can pick from the stuff we studied through. But I want you to concentrate on looking at comparing and contrasting all of those things we discussed. It'll help you not only in whatever essay question I do ask, but also in the other questions you're asked. You know, we talked a lot about how photosynthesis and respiration are the same. Right? There's similarities. We also talked a lot about how they were different. We did the same thing in mitosis and meiosis, and I showed you a whole bunch of slides where we compared them together. And I didn't talk about the slides very long, right? I wanted you guys to go back and look through those slides and use those as a study aid. I wanted you to use those slides so you can study the differences. I do the compare and contrast questions not only just because I want you to do compare and contrast questions, but that's a good way to learn something, especially in biology, all right? Especially in any of the sciences. All right. Does anybody have any questions for me? Specific questions. I don't like to stand up and do a review and basically just talk about things because I don't know what y'all need me to talk about. If you don't need me to talk about something, I don't want to waste your time if it's something that's easily understood. You guys need to give me some idea of what it is you want me to talk about with you. I can give you some little pointers, like for example, let me get a blank whiteboard here, folks. I'll get this off the way. Right there. For example, I told you this in class. If I was going to study the way oxidative phosphorylation works in the mitochondria, remember the part with all the pumps? I would draw it. That's what I go. Okay, this is the phospholipid bilayer. I would get crazy drawing the phospholipids. I might label it I would label it the phospholipid bilayer, something like that, right? And then I would draw the proteins. And I would concentrate on the pumps, because I said there were helper proteins in between, and if you memorize those, that's fine. But I would draw them, and I would say, oh, these are proteins that pump protons across the membrane so they can flow back through ATP synthase. See, by the way, I'm labeling all this stuff as I go along, and here I make ATP. Oh, that's right. And of course, pumps need energy, and where I'm getting that energy is from NADH, FADH2. These are high electron carriers, and they're dumping electrons. FADH2 technically dumps its electron a little bit further down the road, but that doesn't matter. It's dumping high energy electrons into this pump. The pumps are being turned on. They're pumping protons. See what I mean? So do it. Draw that stuff out. And you can make a similar drawing for the one in photosynthesis too. Now again, don't bother to go and memorize all the molecules that are created in the Krebs cycle or the dark reactions, the Calvin-Benson cycle. I would like you to know what's going on during those cycles, what's the purpose, what's being gathered, what's being used. And it's specifically the Calvin-Benson cycle. Pay attention to the numbers of ATPs and NADPHs. We talked about that because that is the reason why we have cyclic photophosphorylation, right? You run out of ATP first. So make sure you do understand those numbers and what that means. All right. Good question. Good question. All right. Again, that's something I would draw out, all right? And I would say, okay, and I'm going to draw it condensed. You could draw it uncondensed. Okay, here it is. It's uncondensed. Now it's condensed, and that's a chromosome. Again, if you're having troubles getting with the chromosome and chromatid, think about the term chromosome refers to technically a set of unique information. So again, if you turned in your buddy's paper for a humanities class, you plagiarized and simply copied it on the copier, there's no unique information there. So when I turn a chromosome into a chromatid, how do I turn a chromosome into a chromatid? What does a chromosome have to do to be a chromatid? Split. Give me a better word. Thank you. Hold on, Kieran. Actually, no. Yeah. Pairing is that can be confusing with homologous chromosomes pairing. So, Sadie, they don't split. What do they do? Trying to divide. Thank you. All right. So there's a chromosome. I even drew a centromere. So then I'm going to try to recreate. Why did I do this? I'm such a bad artist. There we go. Centromere right about in the right same spot. Okay. So I put them together, and of course, what they're being held together with in the middle are cohesion proteins, right? Sticky things holding them together. All right. So now I've got a pair of chromatids, a pair of sister chromatids, but I have a chromosome. All right. Chromosome is a unique set of information. Now, as far as the other structures, you remember the centromere is the narrowing part. It's not always in the middle, but there's a protein attached to it called a connectochore. On each side. And each connectochore is connected to a connectochore microtubule, isn't it? Spindle fiber. And when the right time comes, and this gets broken up, these cohesion proteins, these spindle fibers start yanking these now, as soon as they split, chromosomes to opposite ends. So an organism has 12 chromosomes. Oh, let's do a little exercise here. All right. An organism has the diploid number of 12 chromosomes. All right, let's do a little drill here with the time space. It's got 12 chromosomes. Diploid number is 12. Your diploid number is 46. Its diploid number is 12. How many chromosomes will exist in its gametes? Think of the word. Gametes are haploid. Remember what happens in meiosis. Everything splits right down the middle. Okay? Back to the diploid cell for a second. During mitosis, during metaphase of mitosis, how many chromosomes is that cell going to have? In metaphase of mitosis, how many chromosomes? Shaniqua, how many chromosomes? Jasmine, how about you? How many chromosomes? During metaphase, how many chromosomes is that cell of 12 chromosomes going to have? I guarantee you'll see questions like this on the exam. How many chromosomes? How many chromosomes? Someone help Shaniqua out. How many chromosomes are you going to see during metaphase? I'm going to see 12 chromosomes. How many chromatids am I going to see during metaphase? There you go. All right, now let's jump it up to the next level. Let's take it into meiosis. So here's a cell, 12 chromosomes, right? Diploid number is 12. During metaphase 1, how many chromosomes am I going to see? How many chromatids am I going to see? How many chromosomes am I going to see in metaphase 2? Chromosomes in metaphase 2. Once you've gone through metaphase 1, you've already done reduction division, haven't you? 6. How many chromatids am I going to see in metaphase 2? There you go. There you go. Does that help? You want to do another one of those? Does that help with your understanding? Okay, let's do another one. All right, diploid number. Fill out your calculators here, folks. 38. You know what? Let's do humans. 46. How many chromosomes in metaphase 1? How many chromatids in metaphase 1? Listen to the words I'm saying. How many chromatids in metaphase 1? Okay. How many chromosomes in metaphase 2? And how many chromatids in metaphase 2? And how many chromatids in telophase 2? How many chromatids in telophase 2? In mitosis, how many chromatids in telophase? How many chromatids? So if this were a, we're talking now mitosis, just take this cell. How many chromatids are there going to be in telophase? You thinking it's a trick question? Look in your book. During telophase in mitosis, or during telophase 1 or 2 during meiosis, do you see any chromatids at all? No. So the answer is 0. You don't have chromatids in telophase. You don't have chromatids until they're attached to their copy, which starts in prophase, exists during metaphase, and ends during anaphase. Okay? All right. What other questions? Good. True or false? Everybody does glycolysis. All living organisms do all cells do glycolysis. Yep. True or false? All cells do oxidative phosphorylation. What organelle do you need in your cell in order to do oxidative phosphorylation? What organelle in the cell do you need that in order to do oxidative phosphorylation? The thing I drew just earlier with the pumps. There you go. What does it mean to phosphorylize something? In a very basic sense. Add a phosphate to it, right? Give me an example of something that gets phosphorylized, Marie. Write it on the screen or type it in. Give me an example of a phosphorylation process. Yeah, it does, right? During the light reactions. When we turn ADP into ATP, anytime this happens, this is an example of phosphorylation. Now remember, when we're looking at some of our other molecules, notice I told you like in glycolysis, phosphates sometimes get transferred to a molecule. So that's phosphorylation. It's not of ADP, but anytime you stick a phosphate on something, you phosphorylize it by definition. Yes, absolutely would be, of course, because the word chemiosmosis, and it's kind of an unfortunate word because it makes you think of water, but basically chemiosmosis refers to that ATP synthase and the fact that the way it works is because of diffusion. More here than here, right? So that's the way. Diffusion or osmosis is diffusion, remember? It's just diffusion of water molecules. I wish they'd kind of use maybe the word chemi diffusion here would have been better because there really is no water involved as far as that goes. But yes, that's right, it would be safe. All right, any other questions for me, folks? Got a few more minutes left. Make sure you know the images of mitosis that we studied in class. So go online. Gosh, I could give you a bunch of them right now before we even leave. I could give you a link or two if you want. Allium root mitosis. I've got ones that are even. I think I even post them on Blackboard if I remember correctly. But look at this one. This is a good one here. This one has not only the stages of mitosis, but they're labeled. What? I don't know whose phone number that is. Anyway. call that number. I don't know who that is. Yeah. There's ones for plants too. And there's dozens and dozens of those kind of things. So try them out, okay? Make sure you not only know the different stages, but if I point to something, like I might point to a spindle fiber if it's real obvious, or an aster. I definitely point to an aster, or I might point to the cell plate, or I might point to a chromatid or a chromosome, right? So make sure you not only know what the stages look like, but know what the structures are in the stages. The ones you can see anyway, all right? Any questions on any of that? Where are we? We got a couple more minutes. You guys have any more questions? If not, then by all means, you're welcome to leave. And I'm around in office hours. If you do have questions later on, stop on in and say hello. Other than that, have a good weekend. And I will see you guys on Monday. Don't be late. Make sure you get your assignments done.